Well, thank you, Lars, for that nice introduction, and thank you all for coming. It's nice to see a very nice flock. I stole the line from my neighbor right now. As Lars was saying, I have actually been here at KITP since January, helping organize a program on what we call active matter. And I normally live in Syracuse. Syracuse is nice, but I have to say Santa Barbara has been quite a nice experience, especially this winter, because I've heard a lot from my husband who was back in Syracuse about the winter this year, I have to say. So what I have slightly changed the title here, putting sort of this active matter. Okay, and I already did this. Wait a second. Putting this active matter at the top, which is actually the title sort of our program. And I want to spend a little bit of time telling you what we mean by active matter. But an example is here, is this sort of vortex of tuna, is an example of the many remarkable and sometimes stunning organization of living creatures that we actually see in nature on very large scales. And I will show several examples of these. But the idea here is to try to figure out, I would hope to be able to convince you, that physics can actually play a role in trying to understand this kind of organization on many, many scales, from flock of birds or schools of fish down to things that happen in the interior of living cells. And in particular, what I'm hoping to try to do is to convince you that we can use the kind of ideas that we've been using for a long time successfully to actually describe organization of non-living matter, to describe how materials form, to describe their properties, to describe what we call phase transition, the change of materials from one state to the other, say liquid to water or to water to ice or so on. We can use similar ideas to describe a really broad range of phenomena in this class. So I will start by showing a few examples. Some might be familiar. Quite a few movies. After all, we're here to have some fun tonight. And then I'll give you some specific examples of the properties we look at and try to give you a flavor of how we may go about describing them. So here, of course, you've already seen. Here is a flock of starlings. I picked this picture because this is over the skies of Rome. After all, I'm from Italy. You see St. Peter down there. And you see here is actually Santa Maria degli Angeli, one of the many, many churches in Rome. But the idea is that you see this flock of starlings moving around as a cohesive, soft object. Starlings spontaneously pick a particular direction which to fly, and then they spontaneously changes. It sort of looks like a soft body that behaves collectively as a single entity. Second example, it's a little bit maybe less familiar to you. These are emperor penguins in the Antarctica. A physicist from Germany, Ben Fabry, has actually been studying the huddling, as it's called, of penguins. Emperor penguins, temperature in Antarctica during actually the season where they incubate their eggs goes down to minus 50 degrees centigrade, very different from Santa Barbara. So they need to keep a little warm. And so what they do, they form these packs. But what is really interesting, and I'll show you this other movie, which is sort of an inset. What is really interesting here is that once in a while, a penguin moves, and the motion can be as small as moving their feet of a couple of centimeters. This motion creates a wave that you see up here, and the wave serves to compactify the pack, make it more compact so that the penguins can stay warmer. So another example of collective organization in nature that arises, again, seemingly without a leader, spontaneously, that we would like to try to understand. The next example, we're going down to quite a different scale. These are skin cells crawling on a substrate in an in vitro experiment, which is called a wound healing assay, because it's intended to mimic how cells move when actually we have a wound in our skin. We have a cut in our skin. Cells actually have to move to fill in the region. And this is on an artificial substrate. This was started by sort of packing cells in a narrow region and then letting them go and see how the cell collectively crawl or migrate to fill the wound. Now, the understanding of collective cell migration, collective cell motion, is actually a very important topic because it's not only relevant to wound healing, of course, it's also relevant to things like morphogenesis, embryogenesis, as well as cancer metastasis. How do cells leave the tumor to actually invade the rest of the body? So it's something we are very interested in trying to understand. So this is an example on a very different scale. You see the arrow bar over there is 550 micron. 50 micron is about 1/500th of an inch. Next slide, another example. Let's see. Why doesn't it move anymore? OK. These are bacteria. These are E. coli. E. coli, you've heard lots of E. coli. E. coli are bacteria that have a body which is about 2 to 5 micron long, a tail made of flagella, and they swim around in a fluid, in water, say. But at a high enough density, they form these, again, flocking, if you like, type of patterns. They have some collective dynamics. And at even denser pattern, they actually start swirling around like sort of a turbulent fluid. And again, what drives these collective dynamics is something, again, we like to try to understand. And finally, I think I have sort of just one more. This is actually, from looking at these natural phenomena, physicists and engineers and chemists have taken inspiration to actually build sort of micromotors that are self-powered, that is, little mini machines which may be on the size, this particular rod I'll be looking at here is itself of the size of a few microns, 2 to 5 microns, sort of like the E. coli. They have been building these little particles that really are self-powered, and as we will see in a minute, can actually swim around just like the real bacteria. The first example was actually proposed by a group at Penn State a few years ago, and it was obtained by coating this rod with platinum on one side and gold on the other side. Then when these rods are in a solution and you add hydrogen peroxide to the solution, the platinum serves a catalyst to actually initiate the dissociation of hydrogen peroxide into water and oxygen, and essentially creating gradients of concentration of oxygen that propel this little rod forward. And here is another little movie of these chemically powdered rods or colloids. And as you see, they swim around, not unlike the E. coli I had on the previous figure. More recently, the same group has actually been able to build even smaller rods that are not powered by chemical reactions that are toxic, but are powered actually by ultrasound waves. And they have been able to put them inside cells with the idea here that one is trying to construct mini machines that you might be able to use someday for drug delivery or something of the kind. So again, there is actually nowadays an entire class of these mini machines, and we are studying their collective behavior, which is very similar to the collective behavior of the cell-propelled entities you see in nature. So in fact, I like to stress this similarity. I want to propose a quiz. So these are two movies. I will show them to you. One are bacteria. One are actually these platinum gold cell-propelled rods. And we'll take a vote. You'll tell me which is which. So let's have them start. OK. So let's see. How many people think this is, say, the bacteria? OK. How many people think this is the bacteria? Well, it's about equal. Some people don't refuse to vote. OK. Well, this is the bacteria, and these are the microrods. Actually, two of my colleagues earlier today, when they were tested, they both got it wrong. So it's really essentially impossible to tell. And it's also, when we look at the collective behavior, and this is what motivates us, if you like, to use this artificially powered system to actually mimic some of the collective behavior we see in nature. So these are sort of the examples. And so the point I'm trying to make here is that we see these really broad class of systems, all very different, all on very different length scales, that's for sure, from the birds, the fish, down to the bacteria, the cell. And what do they have in common? Well, they're all systems of many entities, of many particles. I'm going to call them particles. I'm going to call a bird a particle. I'm going to call a fish a particle, because that's what physicists like to do. So they're systems of many particles, but these particles are self-powered. They are able of independent, self-sustained motion. So that's the first thing they all have in common. They clearly communicate or interact with each other in different ways, and they organize in these large-scale structures that we've seen a few examples of. There are also collections of micro-robots that also organize in similar structures. So they're all different, they're on different scales, and each one is governed by different interactions, yet they all exhibit this organized motion at large scale. And the point I would like to stress is that they exhibit this organized motion spontaneously, without a leader. There are some cases in which a leader is needed, but the examples I've shown you are all examples where this motion occurs without a leader, spontaneously. And that's really the key point here. That's really what we would like to understand. So once again, we see these, physicists call this emergent behavior, this organization at large scale. And we see it on scales from the giant flock of birds to, as I will show you a little later, the interior of cells. And the question is, can physics actually say something useful, hopefully something quantitative about this, that then will enable us to actually mimic some of this behavior with our micro machine and reproduce it in artificial systems. On a similar note, I like to make an analogy here with our understanding of states of matter. We know that matter is made of atoms, and we know what are the simplest state of matters that we study in school. You know, we can go from a gas, and atoms interact with each other. If you decrease the temperature or also increase the energy, as a matter of fact, you can go from a gas where the atoms are filling a container and very loosely interacting, to a liquid, to a solid, possibly one here with crystalline order. And the changes from one state to the other are what we call phase transitions, and occur spontaneously as I decrease the temperature or increase the density. The atoms suddenly decide, I decrease the temperature, and the temperature becomes lower compared to the relative interaction, and the gas condensates and spontaneously forms a liquid. So now I have these collectives of interacting, self-propelled entities, like the bacteria or cells or synthetic swimmers or birds. So can we think of them as a new kind of active matter? Again, the key difference is that the particles are self-propelled, but there'll be interactions. I have to figure out what I need to tune equivalent to temperature, where density I certainly can tune, but what is equivalent to temperature that I need to tune, that might lead me to different states, different phases of this active matter. I like to understand what new states are possible and what are their properties. And this is what we have been working on here at KITP during the last few months, since January, in this program that actually will be over in, as a matter of fact, just 2 more days. So what I'm going to do now is sort of give you 3 examples of unusual behavior and try to tell you how we go about trying to model it, trying to explain it. And so the first one, we go back to flocks in a minute. I'll actually describe to you a model we used that was actually initiated to really describe bird flocking. But before doing that, I want to tell you about something that I call surprises in active gases. So there are some unusual phenomena that arise once you make the entities or the particles self-propelled, even at low density, even for a system as simple as what we like to call a gas. And what do we mean by a gas? By a gas, we mean a collection of particles that are relatively weakly interacting with each other. So even in that case, you get something quite interesting. And to understand that, we need to compare the microscopic dynamics of atoms in a gas to the microscopic dynamics of my self-propelled entities, let's say E. coli, for instance. So what these two images here show, here on the left, is what we call a colloidal gas. These are actually, say, plastic beads, maybe 1 micron in diameter, which are in a fluid. There is no flow. The system is sitting there in equilibrium. This is actually an image by Eric Dufresne, Yale, which is here for the program. And what these beads are doing, they're jiggling around with random motion around some mean position. And this jiggling comes about because of the collisions they experience all the time from the surrounding fluid. If we look at the air in this room, the atoms, if we were to look at it under a suitable microscope, we will see that the atoms are not immobile. They're jiggling around all the time. They're undergoing this kind of random motion. But these are small kicks. At every step, the particle goes in a new random direction. The dynamics of something like a single E. coli is quite different. E. coli swims so here is E. coli. So you can kind of see the body and the tail. E. coli swims in a straight line for a while, then spreads out the tail and makes a change in direction. We call this motion run and tumble. And what is important is that that straight line motion, the run, persists for a while before the particle runs. So it's really quite different from this, although you could also say it's a rather subtle difference. And my point I want to make is that this relatively subtle difference in the microscopic dynamics is going to make a big difference at the macroscopic level. So here is this random jiggling motion, and here is this sort of directed motion in a straight line with then a change in direction. The third movie, which I'll run in a minute, that I have down here, is actually very interesting, because these little spots you can barely see here are sort of glass beads, half a micron in diameter. And this is an example of a man-made system made to mimic E. coli. These are sort of like gold platinum rod, only they're a little different. They're actually spherical beads. They have a little magnet embedded in them. And the fluid in which they sit also contains hydrogen peroxide. Sorry, and they sit in a fluid. And when you shine light on them, this magnet embedded in the bead actually creates chemical reactions that make the bead self-propelled. So when I run the movie, you're going to see, the movie is going to tell you when the light is off and when the light is on. When the light is off, the bead will be just like my bead sitting at equilibrium in a fluid. But when the light is on, my bead will swim around just like an E. coli. So that's what you're supposed to be watching for. And let me show that to you. So you see now the light is off, and they're just jiggling around a little bit. Now the light goes on, and they do this run, this straight line motion. The way they change direction is a little different because they don't have a tail. The change in direction is actually due to the random collisions of the fluid, but they do so they change direction more gradually. But you see, again, still you see that the dynamics when the light is on and they become what we call self-propelled particles is sort of similar to that of E. coli and not to that of an equilibrium bead in a fluid. So again, this seems like a rather innocuous difference in the microscopic dynamics, but I'm going to show you makes a big difference at the macroscopic level. One thing we know about gases quite well is that gases spontaneously fill a container. That's good. Otherwise, if we had all the air in a corner in this room, that wouldn't work very well. So the gases we're familiar with do this. Active gases don't. They huddle. Remember our penguins. And active gases are sort of like the penguins. Now, these are the same light-activated beads I showed you a minute before, but now many more of them. It's a slightly denser gas. And let's see what they do when they become self-propelled. When the light is on, they form these little crystals, these clusters. So they huddle. And when the light is turned off, which will be in a minute, in a few seconds actually, you will see these clusters falling apart. So this is a very counterintuitive behavior because it's not due to interactions that might attract the particles to each other. It's only due to the fact that these particles are self-propelled. Let's try to understand it. You can actually understand it with a very, very simple model. Suppose we think now the model is in 2 dimensions, so these are disks, these are not balls, but I will call them billiard balls. Suppose you had billiard balls that are self-propelled. That is, that they have a dynamics that I can prescribe with some little math, not even too difficult, some dynamics that says that they go in a straight line for a while, then change direction, go in a straight line for another while, and so on, sort of like my E. coli. When they can also, like billiard balls, they can collide with each other and repel each other. They push each other apart. That's all there is in the problem. So what happens if I define the dynamics of these billiard balls this way, and I look at their dynamics, put them on a computer, and what happens is that these billiard balls will actually, again, huddle together like the penguins in a big cluster. It's not a static clusters because they're actually kind of trying to get out. They are actually self-propelled particles. So it's not really a crystal. It's still an active system. It has this in motion. But they certainly don't behave like an equilibrium gas that fills the container. And this is essentially because if two of these hit each other, and then before, remember, they travel in a straight line for a while, and then eventually they turn. But if they hit each other, they will be stuck for a little while because they're not yet ready to turn, and other ones may come in over that time scale, and that seeds the formation of this bigger cluster. And in fact, active gas also don't fill the container. They go to the walls. And bacteria are well known to do that, as a matter of fact. They tend to aggregate to the walls. Bacteria actually do aggregate to the walls. They aggregate to substrates. They form solid states called biofilms, which are usually very bad, and we would really like to understand how to get rid of them. So behavior of active gases is quite strange, and it's all due to the fact of these, let me call it again, run and tumble type of dynamics as opposed to the dynamics of a familiar gas. Even more remarkable, these ideas, the fact that the bacteria have these dynamics, means that they interact very differently with walls of certain shape. A dead particle will rattle around if it hits a wall, but a bacteria will actually be guided by the wall for a little while. And this has suggested to physicists that the right shape, this is like a microgear. This is a simulation of a bath of bacteria with a microgear in the middle, which is actually free to rotate. And you see in this simulation that the idea is that the bacteria may be able to power this microgear, but this is not a dream. This was an experiment to go with it. And so bacteria can actually power microgear. This gear itself has a 50 micron diameter. The bacteria are just a few micron. Now, you maybe could ask me about the efficiency of these, and we're not quite there yet to make this useful, but it's tantalizing, the idea that because of these unusual dynamics, you can actually get this effect. If you were to put this in an equilibrium gas, this just wouldn't happen. Your gear will not rotate coherently in the same direction for a little while. So these are the active gases. Now, let's move on and go back to the flocking. So what I want to do now is to show you how we are starting to understand this flocking, indeed, as a phase transition. And again, by flocking here, what I mean is this spontaneous organized motion at large scale without a leader and without applying an external force. If you have a model in physics, that we call the Vicek model, which is the sort of canonical model of flocking. Tamás Vicek, shown here, is a Hungarian physicist who developed this model precisely with the idea of seeing if you could use physics to describe the flocking of birds back in 1995. And so we call it the Vicek model of flocking, and I'll tell you what it is in a minute. But in fairness, Craig Reynolds, a computer scientist working for the animation industry, had actually developed the same model and published it back in 1987. Craig Reynolds, so this is the kind of model that in the animation industry is used to do things like the wildebeest stampede in The Lion King. And Craig Reynolds himself worked in Batman Returns, has won at least one Oscar. But in spite of all of these, physicists are pretty arrogant, so we call this the Vicek model. That's what we do. So it's a Vicek model, but it should really be the Reynolds model. Now, Vicek was a physicist, a condensed matter physicist. He had worked on materials. And so looking at the birds, he was inspired by what he saw as an analogy between the flocking of birds and magnetic properties of materials. Now, we know that we understand magnetic properties of materials actually pretty well. We know that essentially arise because many atoms effectively carry sort of like a little magnetic moment that we call spin, and we describe as a little arrow. So here is my atom with a little arrow. Now, this is now a material, a solid material, with the atoms, and each one carries a spin, carries a little arrow, a little magnetic moment. And generally, at a high enough temperature, I think my pointer is dying on me, so I will switch. At high enough temperature, these little spins point in all random direction, and this material has no special magnetic properties. But if you lower the temperature, what happens is that spontaneously, because of the interaction between the spins, this system undergoes a phase transition, and the spins order, they all point in the same direction, and now you get a magnet, a permanent magnet. Not all materials do this, but some do, and we understand this fairly well. So Vicek thought that if you actually take well, this was done actually after Vicek, but if you take a flock of birds and look at how they are moving and assign a vector, which you can see very well here, but draw a vector for the velocity for each of the birds, you'll get a collective set of arrows that reminds you very much, they're all pointing on average in the same direction, and reminds you very much of the spin of the material, all pointing in the same direction because they've undergone this phase transition. So inspired by this analogy, Vicek proposed a model of, let's call them flying spins, where each bird is a spin, but the spin is not a magnet now. The spin is a velocity, or it has actually, each bird is assumed to have, in the simplest model, a fixed speed. And so really all it counts is the direction of the arrow. So birds are like flying spins. Then we need to figure out some rule, how do we describe the interaction among the birds? In the magnet, it is the interaction among the spins that lead to the formation of the ordered state where all the spins are pointing in the same direction. So we have to specify some kind of effective interactions. And the effective interaction that Vicek specified are the following. He said, OK, let me take my bird. They're all the same, but I just colored one black to point to one of them. And at every time step, my bird looks at all its neighbors within a circle of a fixed radius that I prescribed and aligns with them. So in this particular case, they're mostly going the other way, so it actually switches over and goes the other way. And then takes a step forward in the new direction. However, birds are not perfect. They make mistakes. And so the alignment is not perfect. There is some noise. So they align with the neighbors with some mistakes. And this noise is sort of like the temperature in a real material, in a condensed matter system. So there are sort of two important things in this model that will control the collective behavior of the birds: the density of birds, the number of birds, how tightly packed they are, and this noise that acts like a temperature. And what happens if you write down the math that goes with this model, and you can actually see what it does, is that indeed you get a phase transition from as you tune the noise, say, or the mistakes the birds make. So when they make lots of mistakes, a very high noise, they are all flying in different directions. It's a disordered system. It's a gas. When they make very few mistakes, low noise, you get this coherent motion in one direction. You can't quite see the arrows here, but all the arrows in one region are pointing in the same direction. That's an ordered flock. And actually, we also get these interesting intermediate states where the birds organize in these traveling density waves, not unlike your the density waves you get in traffic jams, as a matter of fact, that here in California you're all very well familiar with, I am sure. So what we get is order without a leader. This occurs spontaneously. We don't have a leader. The birds are all equal. We don't have an externally applied magnetic field or something that tells the birds which direction to go. They spontaneously choose a direction. And in fact, they could have chosen this direction or that direction. It would be the same as far as these models goes. So we get order without a leader out of this very simple model. Of course, there is still a challenge that people are actually working on. The interactions I described to you are at best clearly some kind of effective interaction that I, as a physicist, prescribe for the bird. Can I actually relate them to actual properties of the bird? And people are working on this. I don't have that anymore. Now, after Vycek developed this model, people, a group in Rome in particular, actually a group in Europe, actually, based in Rome, but was a European group called the Starflag Collaboration, actually started collecting an enormous amount of three-dimensional data on flocks by using cameras and making movies of flocks, and then trying to analyze these movies on a computer to reproduce trajectories with the idea of trying to see if the Vycek model could actually be used to fit real data, field data for actually bird flocks. Now, this was actually a very challenging computational task because, of course, what you need to do, you have a movie, you have a frame here with a flock, you have the frame at the next time step. In order to draw trajectories, you need to know which bird went where. And they had to develop very sophisticated algorithms to figure that out, but they did. And there came a surprise. What they found is actually that the Vycek model actually could be used to fit real bird flocks, but with a twist, with a change. They study starlings. So actually, I shouldn't say bird, I should say starlings, because it's far from clear the starlings that other birds would do the same. But they found that in order to use the model, what essentially they found is that the starlings does not interact with all the birds within a circle of a fixed radius. Rather, instead, it interacts always with the 6 or 7 closest birds. Now, at high density, this might not make a big difference, but at lower density, and especially near the boundary of the flock, this actually can make a big difference, as I have tried to kind of show in this little cartoon over here. And so the idea then is that this type of interaction, which physicists call topological interactions, just because they like nice words like that, but this kind of interactions with the fixed number of neighbors actually clearly become important near the boundaries, or where on any time when the density is changing rapidly, and might actually give you more robustness to the flock, the ability of the flock to reorganize more rapidly, which actually may be important, for instance, when a predator is around or so on. So this is this is a model. What I described to you is sort of the simplest model of the simplest variant on what we call the Vycek model. There have been many, many variations, or to try to describe more of the kind of phenomenon I told you about at the beginning, as well as we also have sort of translated this model from a model of particles interacting with each other, which usually we simulate on a computer, to sort of continuum equations, sort of like the equation of flow dynamics, although different because they describe a different behavior. And what these equations have that is different is that they incorporate sort of the energy input that is present in these systems at the local level due to the fact that each of the particles of the bird is actually a self-propelled entity. And with this kind of models, we've been very successful at describing a number of systems. And I want to give you a couple of examples of the other systems we've been describing. So that was the bird. Now let's go to a completely different scale. Let's go to cells. These are individual cells. Well, almost. This is a cell that is dividing, so it'll pretty soon be 2 cells. Now, the point I want to make here, so I've been talking about systems composed of many interacting units. And now I show you a single cell. But the point I want to make is that the interior of the cell is an active material. The interior of the cell is itself, as I'll show you in a minute, composed of many interacting things, proteins and proteins of various types. And it's itself an active material. And it is this active material that drives phenomena such as cell crawling, motion of cells on substrate, or cell divisions, which are clearly phenomena that have a very important mechanical component. And so where physics may hope to be able to make a contribution to understand this kind of phenomena. So let's look at this inside the cell. I said that we think of the cell itself as an active material, so let's look at what are the ingredients of this active material. Let's look inside the cell. This is a cell stained, where in various parts are stained with different colors. Of course, you see here the nucleus. And then you see this kind of network, mostly green with a little bit of red here. This is what we call a polymer network in physics. What it really is a collection of very long filaments or filamentary proteins, which have names such as actin and microtubules. They're very long filamentary proteins linked together by actually a very large number of different small proteins. And altogether, this network, which is called the cytoskeleton, actually serves as both the cell skeleton and the muscle of the cell. It plays an important role in controlling cell motility and cell mechanics, how cells respond, for instance, to applied forces. Now, it's not, unlike our skeleton, is not a static object. It's an object, it's a network that is continuously remodeling. And this remodeling comes from a variety of active processes inside the cell, but in particular, it comes from a class of small protein called motor protein. And this movie, which is running in a loop, is showing you an example of one of these motor proteins. Motor proteins have names like kinesin and myosin. So here is one of the filaments of the network called the microtubule. Here is a small motor protein, kinesin, which, as you can see, is walking on the microtubule. Motor proteins are literally micro machines, molecular machines. They are powered by a chemical reaction, the hydrolysis of something called ATP or adenosine triphosphate. And they're capable of converting chemical energy into mechanical work, into a change in shape, and literally walk on the filaments that compose this network. This thing that you see over here, this, of course, is a rendering. This is not real. But actually, kinesins are imaged, and they look remarkably like that. Okay? They do have these funny steps. So the microtubule, kinesin walks on the microtubule sort of like that. I can't do it properly. I try to imitate my friend Jenny Ross from UMass Amherst. She's very good. But there's a particular microtubule walk that she does. But then she really could have a future as a stand-up comedian too, so not just as a physicist. So this thing that you see here that I call the cargo is a vesicle filled with chemicals, because one of the roles of this motor protein inside the cell is to actually carry chemicals from one end of the cell to the other. But another role of these motor proteins is to form clusters, say 2 motor proteins linked together Then they actually cross-link, say, 2 microtubules, put feet on one, feet on the other. And by working on the 2 microtubules, they actually remodel continuously the network. And these kind of active processes are crucial in controlling some of the behavior, like even cell divisions and cell motility and so on. So it is in this sense, it is this cytoskeleton here, this network of long filaments and motor proteins, that is the active material that drives a lot of the active mechanics of cells. So what we have been doing, I do theory, I do modeling, so I haven't been doing any of these, but my colleagues, experimentalists, have been doing, is to try, this network is actually very complicated. There are many, many different proteins in it. So what they've been doing is extracting a few elements, say microtubules and kinesin, or the other filaments, actin and another protein, putting them in a dish, putting them in vitro, and see if with some very simplified, just a few elements, they can actually reproduce some of the properties of cells, such as spontaneous motion or organization. When the cell divides, this network actually has a very precise way of organizing and splitting in two. So they've been looking at the organization of these cellular ingredients in vitro. And this is something quite remarkable. As I will show in a minute, on the next slide, actually, they actually see flocking at the subcellular level. So what is this picture? What they have done is that they've taken a bunch of these motor proteins, the ones called myosin in this picture, and they have attached them to a glass slide with their feet, which is actually called a head. I think biologists just try to make things confusing for us. But so with their feet, let's call them, sticking up. The feet are the part that binds to the actin and works on the actin. Then they pour over here a suspension that contains a lot of these actin filaments. So now the motor proteins are stuck. They are attached to the slide, but their feet are trying to walk on the filament, and so they will make the filaments move along. The filaments get propelled by the kicking of the motor proteins. And what this will show is, so the filaments of actin can actually be imaged they label fluorescently, and they can be imaged. And you will see that, again, not unlike E. coli, they are doing this sort of zipping motion in sort of random directions, going straight for a while and then changing direction. That's a relatively low density of these actin filaments. When you increase the density, what you see is flocking and density waves, just like we saw in the V-check model. Now here, so you have a dense suspension of these filaments. Only actually 1 in 200 is labeled orange. You see orange on this slide. And when you see bright orange, it means there is a much higher density of filaments there than anywhere else. So I'm just going to show you this again. So again, here you have a flock of actin, which is moving around in a substrate of less dense actin. And here you have these density waves that you saw were traveling in the direction perpendicular to their length. And models, not unlike the V-check model, but with what I would like to call more physical interactions. Models, for instance, where these filaments are thought of as filaments that, yes, are propelled by the motors, but the main interaction may actually simply be that you have so many of that they cannot pass through each other. You know, they repel each other when they are too close. They have a finer size. And so they, like billiard balls, if you like, they repel each other if they come too close. Or interactions that are mediated by the fact that they're sitting in a fluid. Models of this type can actually reproduce both the flocking and the density waves that you've seen in this system. So we are starting to make some progress in actually being able to model and understand some of these spontaneous organization that here is occurring in vitro, but also occurs in a more complicated way inside the cell. And another example I wanted to show you, because I think is really beautiful, this is a similar system This is now a suspension of two other types of filaments and motors that are called microtubules and kinesin. Kinesin was the one that we saw walking along the microtubules. But now it's a suspension where you have clusters of kinesin that can cross-link two microtubules so that they move them relative to each other. When you add the ATP, the fuel that these motor proteins use to actually walk on the filament, so this is sort of a liquid. It's a liquid of filaments, but it's still a liquid. And we are not applying any force. We are not applying any pressure differences. We're not doing anything. We're just putting in the fuel for the motor proteins. And this liquid flows on its own, flows spontaneously. It exhibits these really nice sort of swirling kind of motions, which contains things that we call topological defects that physicists love and probably have nothing to do with biology, but we still like to understand them. And, but the thing that is really remarkable is that this stuff, this liquid, flows on its own. And so we are trying to see whether we can actually understand, say, cell motility as a material property. And actually some progress has been made in that direction. In fact, again, we can actually reproduce this motion very well by writing that by continuum equation, fluid dynamics equations, essentially, but augmented by new terms that model the energy input provided in the system by these motor proteins. These are supposed to mimic, the arrow is supposed to mimic the filaments, and then you see what is highlighted here is the swirling motion that actually comes out from a theoretical model of the kind of microtubule swirling that we saw before. So we really are making some progress towards a quantitative understanding of in vitro systems that at least mimic the function of living systems. Just one more thing, then I will stop. I would like to go back to one of the images I showed you at the beginning, the cells crawling in a what's called the wound healing assay, or these cartoons of cells spilling out from a tumor and invading the tissue in the case of cancer metastasis. This is another system we are actually trying to model, and there have been quite a number of interesting talks here during the program on people making models for this kind of collective cell dynamics. And one question that we are, and actually I should say that Boris Schreman here at KITP is also making a lot of is doing lots of interesting work on this kind of system as well. But one issue we're kind of we're facing here, I made this analogy between dead matter and living matter. One question, we know how to describe solids, we know how to describe liquids, but are tissues liquids or solids? Clearly, there's something in between. They have a liquid-like behavior. I don't know whether this is just an image. I don't remember. It's still the movie. So they clearly have a liquid-like behavior when they sort of spread on a substrate in this wound healing experiment. And actually, you could put an obstacle here, and then the tissue will go around the obstacle, very much like a liquid would. On the other hand, it's pretty clear that there are situations where they behave more like they have elasticity. They behave more like a solid. If nothing else, they support mechanical forces. And so we are still struggling with this dichotomy here and try to make models that will describe the behavior of tissues. But we are making progress. For instance, these are some sort of cartoon-like models. Well, these are models of tissues that are trying to describe, for instance, how individual cells can diffuse through a tissue. And in particular, the idea of this little movie over here, which is actually made by a colleague of mine at Syracuse, was what they have done is that they have put the red cell here is actually a softer cell than the black cells. So the idea is how to see how a cancer cell may move through a healthy tissue. And so we are actually making some progress in this direction as well. So let me try to summarize what I've been trying to tell you and a few things here. So first of all, we can ask, okay, what have we learned so far? Well, certainly we have learned that living matter is a source of beautiful physics and of inspiration for the making of synthetic materials with new functions that might mimic the function we see in living things. But beyond that, I think we really think that physics can actually start to say useful things about the kind of biological organization that I've been describing here on many scales. More specifically, there has been actually some concrete achievements in this field. We have a fairly well-developed theory of the organization and mechanics of living matter that reproduces, at least qualitatively and sometimes more quantitatively, a lot of the organization that I have shown here in a number of different cases. And most importantly, it's a theory that applies on many scales, from giant flocks of birds to the interior of cells. And that is, well, that is what physicists like, first of all, because they like to try to understand phenomena and problems that have some generality when one can define some general laws. But that's also quite remarkable if you think about it a bit. And we have also been able to make a number of systems using chemistry or mechanics that actually imitate living systems. And we are now starting to, particularly physicists and chemists and engineers are now starting to make dense collections of these analog systems and start to get them to work collectively, work together, just like living systems do. Of course, I'm just wanted to mention a couple of important problems. One is the one I just mentioned, a better understanding or quantitative understanding of cell migrations, which is clearly very important for a number of problems like understanding with healing, understanding metastasis in cancer, understanding how to make artificial tissues, for instance. We can understand how it's, and so on. How bacteria at very high density actually form solid-like states called biofilms, which are a problem in many, many cases. That's a field in which many of us are working today. And then again, in getting some actual technology, some real application out of these synthetic imitations people have been building in the lab. So with that, I would like to conclude, and I'd like to thank you all for listening. I would like to thank you, KITP, for its hospitality, and in particular, I really would like to thank the KITP staff, which has been marvelous during the time we spent here running this program. And also all the Active Matter program participants. I think a few of them are here. A lot of them are gone. But I'd like to thank them all for sharing their insights and enthusiasm and for contributing to this talk, whether they know it or not, because I've stolen very freely from many of them without their permission to put in things in this talk. So thanks again.
Questions? Oh, you got it. The microphone's there.
I wanted to see if I understood this exactly. The fundamental premise, then, of the mechanics behind flocking dynamics would be that all physical systems are information systems, and all information systems are structured in layers and governed by rules, which leads me to believe that matter and energy are not fundamental, but information is. So then the question arises, well, what is information? So information seems to be the generating, organizing, directive force behind all physical systems, from biological systems to technological systems. So information, in essence, governs how a system will get built, how it will function, and how it will evolve over time. This leads me to a conclusion that for a physical system to persist in space and time, it must evolve in such a way that it stores, processes, and transfers information in the most efficient manner. So it seems that all emergent behavior, for example, flocking, is governed by the principle of least action, which is a fundamental principle in physics, and Darwinian dynamics. So I think what I conclude is that we seem to have a unifying concept here, unifying anything from biological systems to technological systems. Would that be a correct summation of what you stated?
Well, I think it's a beautiful. So I think information, I didn't mention the word information, but clearly information is very important here because I called it interaction. But certainly, for instance, in the case of birds, one important question is how information is transferred and how actually, for instance, birds react so quick. How the flock, not the individual bird, the individual bird's reaction time is very slow, but the flock reacts so quickly to changes in the environment or something like that. So the transfer of information is a crucial thing here.
Yeah, so it would be the...
Now,
go ahead.
So, we are formulating this way, but I wouldn't say that matter, okay, I'm not sure I remember all of what you said, but I'm not sure that matter, I certainly wouldn't say that the matter or energy are not important because we are also thinking in terms of what I would call physical interactions that certainly involve energy exchanges and transformation of energy from one type of energy to another and so on.
Would you agree that all physical systems then in essence are information systems?
Well,
Okay.
Let me just say one thing, that as arrogant as physicists are, I don't think I would go quite that far. But
Okay.
Oh, hi. I wanted to thank you for a very interesting talk. I wanted to ask just for your reaction to both cellular automata in these models and also random graph theory, connectivity in random graphs. And related to the second, do you know the Erdős-Rényi theorem says that if there's one connection when you reach the level, not 6 or 7 local group, but when you get one, you get superclusters. It's a fundamental theorem in random graphs.
Okay, so your first question was about the cell automata. Okay, some of these models are effectively what one could call cellular automaton. So to me, cellular automaton are models where instead of looking at the dynamics of particles in a continuous way in time, and space, we actually define them as models discrete in space and time. And although I did not say anything about this, some of the models we use here are effectively. But to me, the cellular automaton model is just a way of coarse-graining your problem and defining a model that might actually be computationally more efficient, essentially. Concerning the graphs, I don't actually know a whole lot about random graphs, to tell you the truth. Maybe some of my colleagues here know more than me. But certainly, one thing here that is important is sometimes the interactions among these particles are not the way we like. There might be more hierarchy, especially when there might be more hierarchical than the way we like to think about when we think about, I don't know, a gas or a liquid, where we always think about the interaction as pairwise. Atoms interact with each other 2 by 2. Here there might be more a hierarchical structure where interaction kind of propagate down some kind of a tree, let's call it, or something like that. And so then the kind of ideas you are discussing may actually become important.
Another question?
I had a question specifically about the, I believe it was the microtubules.
Yes.
And the kinesin, the
The
self-sustained
one that flows
flow.
on its own, yes.
Yeah, it seemed like you showed there was flocking and there was density waves.
Yes.
What causes the transition between two
So this is
behaviors?
much denser. This one, the micro. So what closer the transition between? You're talking about the actin then? Okay.
I may be misremembering, but the self-sustained flow experiment.
You talking about this one?
Oh no, these. Yeah. So this is not
okay.
self-sustained flow. Yeah. Thank you.
Well, it is self-sustained motion of some kind, but what causes it? In this case, the transition occurs as a function of either noise or density. You can tune either one, which is very much like a Debye-Check model. There are really two things. So in a material, say you change temperature and you go from, that's the thing we are more familiar with. You change temperature, you go from water to ice. But actually, there are also systems where you can get a transition from a gas to a crystal, from a liquid to a crystal, by just simply changing density. The system becomes denser. Similarly, in these kind of models, you can actually get a transition by changing density or by changing noise. Okay? Noise is like the temperature. In the context of the birds, I called it the mistakes the birds make in aligning with their neighbors. Here is something we kind of put in by hand. Is noise that could come, in this case, from many, many sources. There are variations, these motor proteins, they walk on the filaments, they transform energy from one type to another, but they do it in a way that is not, there is lots of randomness in the system, essentially. So there is lots of noise in the system. And we hide all of our ignorance about all these other processes on smaller scales into this word noise that we put into our mathematics as some kind of a random force. Random force being a force that varies, in some way, in a random way, essentially. So I don't know if...
I just want to clarify. So that's describing the models, and then in terms of those experiments?
In terms of the experiments, it's density.
It's just density.
So yeah, actually, I didn't say. So actually, sorry, in this experiment, sorry, I didn't understand. So this is, I didn't write down the numbers, so I do not remember the values of the density. This is a relatively low density. Then that one is intermediate density. That's the highest density. So in this case, yes. Sorry. Give you a long answer for that.
Hello. So I also have a question about the actin and myosin. And if you scale up to actual coordinated and directional muscle movements, how does this kind of random multi-directional movement correlate to that?
Oh, muscles are very well organized. In muscle, you have something called sarcomere, which are very well organized structures of actin and myosin. And the myosin is the one that causes the ability of muscle to contract and therefore exert forces. But it's a different story. They are organized. So these are random structures, if you like, in the sense that it's like a gas and then spontaneously organizes into these things. Muscle sarcomeres are built to be organized structures. And then they have the ability, so you have these fibers connected by pretty large clusters of hundreds of myosins. And the myosins, by walking on the filament, can pull the muscle fibers together. And so it's a different story. Those are, it's like a crystal. Sarcomere is like a crystal where things are organized in some periodic fashion. No, this would be like taking the muscle apart, taking out the actin and the myosin, and making a soup out of them, okay? And then adding this ATP that fuels the myosin, and that's what you get.
I was wondering if this had any application in cosmology, specifically star formation out of dust clouds.
Oh, I think we're going to ask this to Joe Polchinski, who is sitting right. I think this is a question.
I think the short answer is no. Yeah. Michael.
Thank you for the talk. It reminds me of some work I was involved with in the early 1960s, trying to figure out how humans move about inside supermarkets. We were trying to find out whether you could design a more efficient supermarket, yes, and in particular a more profitable one.
Okay, excellent.
And I wondered whether in the work the team has been doing, they have come upon any interesting stuff about the way humans move about.
Oh, yes, there has been. Actually, maybe I should have included this movie. So, for instance, there are researchers at Cornell that have looked at, use these kind of ideas to model how people move or dance, although I think it's move really, in heavy metal concerts when they are confined to something called a mosh pit or something like that. and they generate, and then their dancing actually becomes sort of this kind of collective rotation, collective motions, and these models have been used to describe this kind of dynamics. I don't know that I would call it an application, but but that's and in other instances as well, like crowd control in stadiums, for instance, and things like that. Yes, so absolutely.
I don't know why I was thinking this, but I was wondering if you looked at populations over huge time scales, world populations and how we move in suburbs and cities and stuff like that, whether that would also exhibit that behavior.
I don't know much about that. I know some people have looked at this, but I'm afraid I don't know very much about that. Yeah, but it's an interesting idea.
If you don't mind, how did this brilliant, bright little girl grow up in Italy to become this extraordinary physicist, and what set it off? Well, how did you veer off into this field?
Well, so it's not easy to answer because, first of all, I spent my high school years studying ancient Greek and Latin and philosophy and art history and things like that. Okay, and although in Italy we also, in all high schools, we also have to do 3 years of physics and mathematics, and so it was towards the end of my high school that I realized I actually really like science, but I was very torn because actually I also like the study of languages from this kind of philological, comparing the roots of words and so on. So anyway, eventually I decided towards the end of my high school that between the two, I was more likely to be able to get a job if I did physics than if I did ancient languages, and so I went in that direction. Now, I then came to the United States to do my PhD, mostly because at that time in Italy there was simply no way of staying at a university to do research. There was no PhD program, so I came to this country. And then kind of a series of, and then why did I stay here? Partly a series of accidents, my husband is also a physicist. I met him in this country. It was easier for us to get to jobs here than elsewhere. But so anyway, to make a long story short, I kind of, I liked very much when I was in school to think about things very logically. But I like this both in the context of science and in the context of other things like languages. And then eventually the decision was made more kind of because I felt, well, maybe in this direction I have more chance of getting a job. But then I really liked it, and so I just stayed with it.
Thank you.
That's great. Let's thank Cristina. So let me remind everyone there's some cold refreshments out near the courtyard. So if you want to flow or flock that way, you may. And we'll open this up.